Hi students, welcome to the first video module of Neet UG 2025 Rapid Revision Series by Impetus Learning. So as we discussed earlier, we will complete our revision of the whole Neet biology within the span of within 20th April, and then you will get adequate time to practice your questions, then test series, then DPPs. Okay. So today myself Debrup, your friend philosopher and guide and also biology faculty of Impetus Learning. तो आज हम शुरू करेंगे द मोस्ट इम्पॉर्टेंट चैप्टर ऑफ द होल नीट सिलेबस एंड यू कैन से लाइक फ्रॉम दिस चैप्टर एवरी ईयर 7 to 8 क्वेश्चंस आर बीइंग आस्क्ड एंड दिस इज कंसिस्टेंट एंड तुम अगर ये चैप्टर कर लोगे दैट विल इंश्योर अराउंड 28 to 30 मार्क्स 28 to 32 marks in the NEET exams. Okay, so let's start with our first video module, principles of inheritance and variation. So basically मैंने क्या किया है, like I have marked your whole NCERT by two colors. One is with green, another one is with red. तो green वाले का क्या होगा, like green वाला is like that conceptual portion, like that is not too much very important from the MCQ purpose, and that has been never asked in the exams, but that might be asked in the NEET 2025 exams. और तुम्हारे कंसेप्ट लगेंगे तुम्हारे क्वेश्चन सॉल्व करने में. और जो रेड वाले में मार्क किया वो सबसे इंपॉर्टेंट टॉपिक्स होंगे. दैट विल बी योर की लाइंस. द डे बिफोर एग्जाम यू विल बी रिवाइजिंग दोज लाइंस एंड द क्वेश्चंस वर बीइंग आस्क्ड डायरेक्टली फ्रॉम दोज लाइंस. ओके, सो लेट्स स्टार्ट. तो द नेम ऑफ द चैप्टर इज इटसेल्फ इनहेरिटेंस एंड वेरिएशन. सो व्हाट इज इनहेरिटेंस? Inheritance is a process by which characters are passed from parent to the progeny. So what is parent? Parent means the parent generation, like the father and the mother. Okay. So what is progeny? Progeny is their next generations. It can be their son, it can be their daughter. Okay. One characteristic trait that is being passed from father and mother to their daughter or son, that is called inheritance. So what is variation? Variation is the degree by which progeny differ from their parents. लाइक हम सब अपने मम्मी पापा जैसे होते, राइट, बट टू सम एक्सटेंट, पूरा तो नहीं होते. तो जितना पार्ट हमारा डिफरेंट होता है तो दैट इज कॉल्ड वेरिएशन. ओके, सो वेरिएशन का पूरा कंसेप्ट छुपा हुआ था इन द वाज हिडन इन द सेक्सुअल रिप्रोडक्शन. तो देखा गया था कि जो जो ऑर्गेनिज्म सेक्सुअल रिप्रोडक्शन करते हैं उनमें वेरिएशन होता है, और जो जो असेक्सुअल रिप्रोडक्शन करते हैं उनमें डिग्री ऑफ वेरिएशन बहुत ही कम होता है. ओके, तो so the most important topic of this chapter is Mendel's law of inheritance. Okay, so who was Mendel? Mendel actually was father of genetics. He was called father of genetics, and these questions were asked many times in NEET exams. Who is known as a father of genetics? So what did he do? He did his experiments on garden peas for 7 years, and the time span is also important. 1856 to 1863. ये सवाल भी नीट में बहुत बार पूछा गया है. व्हाट वाज द टाइम स्पैन ऑफ द मेंडेल्स एक्सपेरिमेंट. ओके, तो ये वाला क्वेश्चन भी तुम्हारे नीट में बहुत बार पूछा गया है. अगला क्या देखेंगे हम कि मेंडल वाज द फर्स्ट पर्सन हु यूज्ड द स्टैटिस्टिकल एनालिसिस एंड द मैथमेटिकल लॉजिक इन हिज एक्सपेरिमेंट. ओके, तो ये वाला क्वेश्चन में पूछा गया है, हु वाज द फर्स्ट to use the statistical analysis and the mathematical logic in his experiments. Okay, तो उन्होंने क्या किया था, let's say he took the garden pea plants. Okay, तो उनका क्या होता है, they have different characteristic traits. Okay, and they are very contrasting, like you can differentiate it by seeing it. मतलब क्या, let's say अगर किसी pea plant का seed shape देखे, right, तो वो क्या होगा, राउंड हो सकता है और रिंकल्ड हो सकता है. You can see here the dominant trait and the recessive trait. So what is a dominant trait? Dominant trait means ये वाला trait is more seen and और recessive वाला trait क्या होगा, that is less seen. Okay, और dominant recessive वाला portion वो allele में हम discuss करेंगे. I am coming to that point. Okay, then comes the seed color. So dominant one is yellow and the green one is recessive. Okay, so flower color is violet is dominant and white is recessive. Okay, then the pod shape, that is full is dominant, constricted is recessive. Okay, pod color green is dominant and yellow is recessive. Okay, so pod color और seed color दोनों opposite होते हैं. seed color में yellow होता है dominant और pod color में green होता है dominant. Okay, flower position axial होगा dominant और terminal होगा recessive. Okay, और stem height में tall होगा तुम्हारा dominant और dwarf होगा तुम्हारा recessive. तो यू शुड बी रिमेम्बरिंग दिस 7 कॉन्ट्रास्टिंग कैरेक्टरिस्टिक्स ऑफ अ पी प्लांट. ओके, दिस क्वेश्चन इज वेरी वेरी इंपॉर्टेंट और ये बहुत बार पूछा गया है नीट के एग्जाम में. ओके तो मेंडल ने क्या किया था, ही यूज्ड टू डू आर्टिफिशियल हाइब्रिडाइजेशन फॉर pollination and he used the cross pollination for his experiments. Okay, तो मेंडल ने क्या किया था, he selected 14 true breeding contrasting trait वाला pea plant. तो this was asked in many times, like what is the number of the plants that were being used by Mendel. So the answer is 14. Okay, you just to remember the number. Okay, now you can see this table. This is very important, which I discussed earlier. That is characteristics, stem height, tall, dwarf. ठीक है, one is dominant, another one is recessive. Okay, flower color violet white, flower position axial terminal, pod shape inflated constricted, pod color green yellow, seed shape yellow wrinkled, seed color yellow green. Seed shape round wrinkled. Okay, so you have to remember this table. Okay, you have to remember this table. Okay, now we will be discussing the inheritance of one gene. तो वन जीन मतलब क्या होता है, लेट्स से कि देयर विल बी वन ट्रेट. ओके, लेट्स से वी कैन टेक लाइक हाइट. ओके, इफ ओनली हाइट इज टेकन कंसिडर्ड एज इनहेरिटेंस, देन दैट इज कॉल्ड द मोनोहाइब्रिड क्रॉस. मतलब अगर एक कैरेक्टर से तुम इनहेरिटेंस जज करोगे तो उसको हम मोनोहाइब्रिड क्रॉस बोलेंगे. अगर मोर देन वन से करेंगे तो उसको हम डाइहाइब्रिड क्रॉस बोलेंगे, और हम वो आगे जाके वीडियो में देखेंगे. ओके, तो क्या हुआ था कि he crossed tall and dwarf pea plants to study the inheritance of one gene. Okay, so plants of the first hybrid generation, this generation is also called the filial one or progeny generation. Okay, तो अगर हम किसी का अगर वो कराते, like let's say hybridization कराते, तो उसका पहला जनरेशन को हम पेरेंट जनरेशन बोलते हैं. ओके, पहले जनरेशन को हम पेरेंट जनरेशन बोलते हैं. ओके, उसके बाद वाले जनरेशन को हम F1 जनरेशन बोलते हैं, उसके बाद वाले जनरेशन को हम F2 जनरेशन बोलते हैं, लाइक फिलियल 1 जनरेशन देन फिलियल 2 जनरेशन. ओके, सो ही फाउंड दैट द F1 ऑलवेज रिजेंबल्ड ईदर वन ऑफ द पेरेंट्स एंड द ट्रेट ऑफ द अदर पेरेंट वाज नॉट सीन इन देम. तो ये क्या हुआ, लाइक लेट्स से तुमने एक टॉल पी प्लांट के साथ एक ड्वार्फ पी प्लांट का तुमने लेट्स से हाइब्रिडाइजेशन करवाया. ओके, तो पहले जनरेशन में क्या होगा, दैट विल बी द ऑल ट्रीज आर टॉल. तो क्या हो रहा है कि वन ट्रेट इज डोमिनेंट ओवर द अदर. सो वन इज कॉल्ड डोमिनेंट ट्रेट, अनदर वन इज कॉल्ड रिसेसिव ट्रेट. ओके, सो व्हाट आई डिस्कस्ड लाइक इन द प्रीवियस चार्ट, लाइक कौन सा डोमिनेंट होगा कौन सा रिसेसिव होगा. ओके, तो ये क्या होगा, इसको हम बोलते हैं लॉ ऑफ डोमिनेंस. तो मेंडल का पहला लॉ होता है लॉ ऑफ डोमिनेंस. इसमें मेंडल ने क्या बोला था, वन कैरेक्टर विल बी डोमिनेंट एंड अनदर वन विल बी रिसेसिव. ओके, हाँ, अब मेंडल ने क्या किया, Mendel then self-pollinated the tall F1 plants. To his surprise, found that in the filial 2 generation, some of the offspring were dwarf. So what happened कि first filial generation में तुम्हारा बस एक ही कैरेक्टर डोमिनेंट हुआ, तो उसको हम डोमिनेंट कैरेक्टर बोलेंगे. उसके बाद जब हम ये हाइब्रिड वाला प्लांट को लेके उनके बीच में जब हम क्या करेंगे, सेल्फ पॉलिनेशन करवाएंगे, देन वी विल सी लाइक देयर आर सम ड्वार्फ प्लांट्स. ओके, there are some dwarf plants. So this shows the second law of Mendel, that is the law of segregation. So what the law of segregation says, it says that the characters which लगता है कि वो फर्स्ट फिलियल जनरेशन में वो सेग्रीगेट नहीं हो पाया, वो गुम गया, बट इन द सेकेंड फिलियल जनरेशन the all characters, it might be dominant or it might be recessive, that segregate from each other. So this is the law of segregation and this is also known as Mendel's second law. तो मेंडल का पहला लॉ क्या हुआ, Mendel's first law is law of dominance, then the second law is Mendel's law, that is law of segregation. Okay, so we will see the monohybrid cross by a chart. Okay, let's see. I said you like there are two alleles in gene. Okay, that is T and T, small t and small t. So T, बड़ा वाला T will just like show the allele which will carry the gene for a tall plant. Okay, and small t is the gene that will is the allele that will carry the gene for the dwarf plant. Okay, and the capital T is dominant over the small t. It means अगर capital T और small t साथ में रहेंगे तो capital T का जो होगा phenotype वही बाहर दिखेगा. That means tall. Okay, so capital T इसको हम बोलते हैं homozygous dominant. Okay, इसको हम बोलते हैं homozygous dominant. That is crossed with a small t, that is a homozygous recessive plant. Okay, so what we can see, there are two gametes. From here there is capital T, and from here that is small t. Okay, then we are getting the first filial generation, that is the capital T small t, and the phenotype of the generation is tall. Okay, then we are doing the selfing, that means we are doing the self pollination. So what we are getting, this is a kind of table, we will discuss it about later. This table is known as Punnett square. This is known as Punnett square. Scientist Punnett discovered this square to show the inheritance of different alleles. So this is called as Punnett square. Okay, तो इसमें क्या दिखा? इसमें दिखा कि there are 3 tall and 1 dwarf plant. Okay, so there is a ratio of phenotypes of 3:1. Okay, so the phenotypic ratio is 3:1 and the genotypic ratio is 1:2:1. इसका मतलब क्या है? जीनोटाइप मतलब है, लेट्स से ये वाला था कैपिटल टी, सो ये हो जाएगा होमोजाइगस डोमिनेंट. ओके, अगर ये कैपिटल टी स्मॉल टी, ये वाला अगर पोर्शन हम देखें, ये वाले पोर्शन को हम बोलेंगे हेटेरोजाइगस. इस वाले को हम क्या बोलेंगे? इस वाले को हम बोलेंगे हेटेरोजाइगस. ओके, हेटेरोजाइगस. और इस छोटे वाले को हम क्या बोलेंगे? इस छोटे वाले को हम बोलेंगे होमोजाइगस रिसेसिव, बिकॉज इसका अलील रिसेसिव है. ओके, और ये होमोजाइगस है. होमोजाइगस मतलब दोनों अलील सेम होंगे, सो दैट इज होमोजाइगस रिसेसिव. ओके, नाउ व्हाट आर द एलील्स? जीन्स विच कोड फॉर अ पेयर ऑफ कंट्रास्टिंग ट्रेट्स, दैट इज नोन एज एलील्स. सो व्हाट आर द एलील्स? एलील्स होते हैं दोनों कंट्रास्टिंग कैरेक्टर को वो इंट्रोड्यूस करते हैं, राइट? दैट इज कैपिटल टी दैट कोड्स फॉर अ टॉल and small t that codes for a small or a dwarf. Okay, as you can see here, capital T is used for the tall trait and the small t is used for the dwarf trait. Okay, and the capital T and the small t alleles of each other, so hence in the plants the pair of alleles for height would be capital T small t and small t small t. Okay, so let's move on to a question, that is a PYQ of NEET 2023. They are saying कि a heterozygous plant with violet color was crossed with a homozygous pea plant with white flower. Okay, so violet is dominant over the white. So what they have said, like, just a second. What they have said? They have said a heterozygous pea plant with violet flowers. Okay, let's say violet ko we can write it as VV. Okay, and they have said a heterozygous, so it will be capital V small v. Okay, and they have said with a homozygous white plant. Okay, and they have said violet is dominant over white. Okay, so what will be the genotype of the white plant? That will be small v and small v. Okay, so what they have said, which one of the following represents the expected combinations among the 40 progenies formed? So we are making gametes from the portions. Here are gametes है capital V and small v. Okay, और यहाँ पे gametes होंगे small v. Okay, so now we will make a Punnett square. Okay, just make a Punnett square that will look like this. Okay, so here will be capital V small v, here will be small v small v. Okay, so it will be capital V small v, it will be small v small v, it will be capital V small v, it will be small v small v. So we can see like ये वाला होगा तुम्हारा tall, sorry, violet, because violet is dominant, right? So in the heterozygous violet is expressed and this will be your white. Okay, so if 40 progenies are produced, right, so 20 will be violet and 20 will be white. Okay, so see what they have told. 30 produced with violet and 10 with white flowers. It is wrong. 20 produced with violet and 20 produced with white flowers. So this will be your answer. Okay, so this will be your answer. Okay. See, this is the same monohybrid cross I discussed in the last page. That is, there is a tall plant, there is a dwarf plant, and we know it like tall trait is dominant over the dwarf trait. Okay, so what they have done, like they have made gametes like tall t, capital T and small t, and that will give capital T small t in the first filial generation. That is the tall hybrid generation, right? That is the tall hybrid generation. But when they are done selfing, that means self pollination, they will produce both tall and dwarf. Okay, so they will produce both tall and dwarf. what will be the genotypic ratio? Genotypic ratio would be 1:2:1 and the phenotypic ratio would be 3:1. This is a high yield topic for NEET MCQ. ये बहुत ही फेवरेट क्वेश्चन है एनटीए का, बहुत बार दे चुके हैं एंड दिस इज अ इम्पॉर्टेंट क्वेश्चन. ओके, नाउ कमिंग टू द नेक्स्ट टॉपिक. What is Punnett square? Punnett square is the diagram that is used for the production of to show the production of gametes and the zygotes in the first filial and second filial plants can be understood from a diagram. So it was a kind schematic diagram, right? Like we make a square and then just we put the gametes over the, like यहाँ पे 2 gamete रखेंगे और नीचे 2 gamete रखेंगे. Okay, so that will give the whole scenario of a pollination results. Okay, so now moving to the next question. See, the production of gametes by the parents, formation of zygotes, the F1, F2 plants can be understood from a diagram called. So what is the diagram called? What we read? That is a Punnett square. That is a Punnett square. Don't forget it. It was asked in NEET 2021. That is the Punnett square. Okay. Another question. The production of gametes by the parents, the formation of zygotes, the F1 and F2 plants can be understood using. What will be the answer? That will be Punnett square. That will be Punnett square. This question was asked in NEET Odisha 2029. Okay. तो हम क्या देखे रहे? हम देखे थे कि in the Punnett square there were 1 out of 4 was homozygous tall, right? 2 out of 4 were heterozygous tall, and 1 out of 4 was homozygous recessive. That means homozygous dwarf. Okay. So like it was like this one. See. Capital T, small t, capital T, small t. That will be capital T, capital T, capital T, small t, capital T, small t, small t, small t. Right? So this one is 1, this one is 2, this one is 1. Okay? So that is 1/4, that is 2/4, that is 1/4. Okay? So they have written the same thing here. So the genotypic ratio will be 1:2:1. And the whole 1/4:1/2:1/4 ratio, it can be mathematically expressed by the binomial expression ax plus by ka whole square. So this was asked in some exams, like by which equation we can express the genotypic ratio of 1:2:1 in a monohybrid cross. That your answer will be ax plus by ka whole square. Okay. So now what is a test cross? Okay. From the name itself you can see we are testing something, right? We are testing something. What we are testing? Let's say एक प्लांट है जिसका टॉल वाला जो ट्रेट है, जिसका जो टॉल वाला ट्रेट है दैट इज डोमिनेंट, राइट? टॉल वाला ट्रेट इज डोमिनेंट, टॉल इज डोमिनेंट. ओके, सो टॉल डोमिनेंट ट्रेट का जीनोटाइप क्या क्या होगा? वन विल बी कैपिटल टी, अनदर विल बी कैपिटल टी स्मॉल टी. माने ये होगा एक होमोजाइगस वाला और ये होगा एक हेटेरोजाइगस वाला, राइट? तो हम कैसे समझे कि टॉल वाला होमोजाइगस है या हेटेरोजाइगस है? तो उसके लिए जो टेस्ट हम करते हैं दैट इज कॉल्ड टेस्ट क्रॉस. सो व्हाट हैपेंस इन टेस्ट क्रॉस? लेट्स से देयर इज अ टॉल प्लांट. ओके, वी डोंट नो, इट माइट बी कैपिटल टी और कैपिटल टी स्मॉल टी. वी जस्ट pollinate it with a homozygous recessive plant. That means a dwarf plant. Okay, that is a dwarf plant, that is small t and small t. Okay, so a tall plant is like crossed with a dwarf plant. So what will be the results? तो इसके 2 results होंगे. Let's say the plant is capital T. तो क्या होगा उसका Punnett square? It will be like this. So capital T, it will be capital T small t. So if you see the results, capital T, so you will see like all the plants here are tall, right? They all are tall. Okay, but if the plant is capital T small t, then what will be the Punnett square? It will be like this. It will be like this. Capital T small t, small t, small t. So there will be a dwarf plant. This wala. This will be your dwarf. Okay, this will be your dwarf. So by crossing a dominant trait wala plant with a recessive trait wala plant, we get some kind of plants in the next generation. That might be all tall, and then that might be all dwarf. That means there are no recessive characters present either, or there is presence of some recessive characters. अगर recessive characters नहीं हुए तो हम समझेंगे that will be homozygous dominant. And if the recessive character is present, like here the dwarf is the recessive character, so we can say easily like this is the heterozygous plant. Okay. See the question. It was asked in NEET 2024. In a plant, black seed color is dominant over white seed color. So they have said like black wala is dominant over the white wala. Okay. So in order to find out the genotype of the black seed plant, like black is dominant, right? तो उसका genotype ढूंढना पड़ेगा कि वो homozygous है या heterozygous है. The genotype of the black seed plant with which we will following genotype we will cross it. So this is known as test cross. This is known as test cross. जिसमें हम क्या जानते हैं कि एक डोमिनेंट कैरेक्टर का जीनोटाइप क्या है, दैट इज होमोजाइगस और हेटेरोजाइगस, राइट. तो व्हाट इज डन इन टेस्ट क्रॉस, वी क्रॉस डोमिनेंट वाला कैरेक्टर विथ ए रिसेसिव वाला कैरेक्टर. सो वी विल फाइंड सम जीनोटाइप दैट कैरीज सम रिसेसिव फिनोटाइप्स. तो हम देखेंगे, ये देखो ये वाला डोमिनेंट होगा तो ये वाला नहीं होगा. ये वाला देखो एक डोमिनेंट है एक रिसेसिव है, एंड बी इज कैपिटल बी इज डोमिनेंट ओवर स्मॉल बी. सो दिस विल नॉट बी द आंसर. This also will not be the answer. What will be your answer? That will be this one, small b. That is, this is the homozygous recessive character. Okay, this is a homozygous recessive character, and we cross the dominant wala trait with the homozygous recessive character. Okay, so small b will your answer. See, this one is the test cross which I explained you in the last page. Like all the flowers are violet means all are dominant. Okay, all are dominant. Hence, what it deduces? It deduces that the plant is homozygous dominant. Okay, and here you see half of the flowers are violet and half of the are white. So here there is a presence of recessive character. So what will be our deduction? That will be the heterozygous. The test plant is a heterozygous genotypic plant. Okay. So what does the law of dominance says? As we read in the Mendels's first law. This is known as the Mendels's first law. This is known as Mendels's first law. Okay. So what does it say? It says कि characters are controlled by a discrete units called factors. Okay. Factors occur in pair. In a dissimilar pair of factors, one member of the pair dominates over the other. So that is the concept of the dominance. Like capital T will be dominant over the small t. Okay. So this is called the law of dominance. So what is law of segregation? It says that the alleles do not show any blending and the both characters are recovered as such in the F2 generation. Like मैंने बोला था कि first filial generation में all plants will be tall and the homozygous and the dominant character will be prevalent. There will be no presence of the recessive character. तो क्या होगा? They will be segregated in the second filial generation. That is your F2 generation. Okay. So this is the law of segregation. Okay. Now these are the some exceptions of the Mendel's law. Okay. So that is one incomplete dominance. So what is the incomplete dominance? Means देखो there is a plant whose dominant trait is red. Okay. The flower is red and the recessive trait is white. So what we do? We do a cross between a dominant and a recessive. So what we get in the first filial generation, like what we expected in the dominant character, that all plant will be red, all flowers will be red, right? But in a plant that is known as snapdragon or antirrhinium species, this was very much frequently asked question in NEET. Give an example of an incomplete dominance. Your answer will be snapdragon or antirrhinium species. Okay. तो इसमें क्या होता है कि a true breeding red flowered plants, that is capital R, and a true breeding white flowered plant, that is small r. The F1 generation was pink. So what is the color of the first generation? That is pink. That is neither red nor white. So what happened here? Like red is not completely dominant over the white. Okay, that is incompletely dominant. So the phenomenon is known as incomplete dominance. The phenomenon is known as incomplete dominance. So what will be the phenotypic ratio? That will be red 1, pink 2, white 4. Okay. So in the second filial generation, the phenotypic ratio will be equal to the genotypic ratio. That is 1:2:1. So this is a frequently asked question in NEET. Like from which phenomenon the phenotypic and the genotypic ratio of the plant will be same in the second filial generation. They were given the options like incomplete dominance, complete dominance, pleiotropy, multiple alleles. So your answer will be incomplete dominance. And the example is snapdragon or antirrhinium species. Okay. So let's say, what is the reason of the incomplete dominance? Okay. So Ncert says there might be a non-functional enzyme and there is a no enzyme at all, and there is a normal character, that is the normal enzyme. If the enzyme is normal, then the normal dominant rule will be applied, okay. But if there is a defect in the enzyme, that will produce the incomplete dominance effect, okay. So you can see easily from the this portion, this diagram, and this chart, okay. You have to remember, okay, key what is the example of a incomplete dominance and how it happens, okay. See, this was asked in the Neet 2024. A pink flowered snapdragon plant was crossed with a red flowered snapdragon plant. What type of phenotypes is or are expected in the progeny? So what they have said? What they have said? They have said a pink flower snapdragon. So what is the genotype of the pink flower snapdragon? That will be capital R, small r. Okay, that is crossed with a red flower snapdragon plant. Red flower snapdragon plant. That is completely dominant. Okay. So what will be the gametes here? Capital R, small r. And what will be the gametes here? Capital R, capital R. Okay. Now we can draw a Punnett square. Okay, like this. So capital R, small r, capital R, capital R. That will give a capital R, capital R, capital R, small r, capital R, capital R, capital R, small r. So this one is red, this one is red, this one is pink. Okay. तो half होगा तुम्हारा red और half होगा तुम्हारा pink. So what they have options given? Only red flower plants. This is wrong. Red flowered as well as pink flowered plants. सही बोला है. Only pink flowered plants. गलत बोला है. Red, pink, white. White भी नहीं होगा. So what will be your answer? Your answer will be red flowered as well as the pink flowered plants. If you want to solve this question, please pause the video and think yourself. Then you can match your answer with me. Okay. See, what is codominance? Okay. So you have to remember codominance means एक साथ, like some trait will be dominant together. So what is the concept? Like if there are two traits, they are not like one is dominant over other, they will equally dominate each other. Okay. So the example is human blood group, that is AB. Okay. So what is the main concept of the AB blood grouping? So ABO blood groups are controlled by the gene I. You have to remember this. This gene I, this controls our blood group. Okay. So the gene I has 3 alleles. So gene I has how many alleles? Just 3 alleles. Okay. That is IA, that codes for A blood group, IB, that codes for the B blood group, and the I, small I, that codes for the O blood group. Okay. So what is the actual blood group? So actual blood group होता है the presence of surface antigen over the RBCs. Okay. If A antigen is present, so it will be A blood group. If B antigen is present, that will be B blood group. If no antigen is present, that will be your O blood group. Okay. So what they say, IA and IB are completely dominant over the small I. Means if in a blood group IA or IO, that means small I is present, the capital IA wala portion will express it. So the blood group of the person will be A. Okay. If IB is present with IO, the blood group of the person will be B. Okay. But when IB, when IA and IB are presented together, they both express each other, right? And what happens if a IA and IB remains together? The blood group of the person will be AB, right? It will not neither be A nor neither be B. It will be AB. So they codominate each other, right? They codominate each other. See, if the allele from parent 1 is IA and allele from the parent 2 is IA, so genotype of the offspring will be IA. So what I said, like the genotype will be IA and the blood type of the offspring will be A. Okay, like this in Neet they can give you many questions solving the blood group issues. Okay, I can find a question for you. Just a second. Okay, let's search it a little bit later. Let's come to the topic. Okay. So actually ABO blood group is also an example of a multiple allele. So what is a multiple allele? There is a presence of more than 2 alleles in a one gene. लेट्स से हम अब तक क्या बोल रहे थे, दैट इज कैपिटल टी कैपिटल टी कैपिटल टी स्मॉल टी, देयर आर 2 alleles, okay, right, capital T and small t. But in the ABO blood group there are 3 alleles. What are the 3 alleles? That is A, that is the B, that is the O. समझे? So this is also an example of a multiple allele. So they have said multiple times कि ABO blood grouping is an example of a multiple allele, codominance, both of the answers, none of the above. So your answer will be both of the above. Okay. So in the Ncert they have said occasionally a single gene product may produce more than one effect. For example, starch synthesis in pea seeds is controlled by one gene. It has 2 alleles, that capital B and small b. Starch is synthesized effectively by capital B, that is homozygous, and the large starch grains are produced. In contrast, small b homozygous have lesser efficiency in starch synthesis and produce smaller starch grains. Okay. After maturation of the seeds, capital B seeds are round and the small b seeds are wrinkled. Heterozygous produces round seeds and seem to be B to be the dominant allele. Okay, but the starch grains produced are of intermediate size in the capital B small b seeds. As you see, another example of a codominance. Okay. Now, inheritance of 2 genes. That means dihybrid cross. Okay. So, what is a dihybrid cross? Basically, dihybrid cross is a cross where 2 characters are used. तो इसको हम समझाने के लिए let's take a fresh page. Okay. So in the monohybrid cross, we took only one character, that is your height, that can be tall, that can be dwarf. Okay, but in this portion we are saying ki like the seed, okay, the seed of pea plant, right, seed of pea plant, that will be RR, that will be round, okay, and smaller, that is wrinkled. Okay, so the round one is dominant over the wrinkled one. Okay, and the yellow, that is capital Y, that is yellow. So yellow one is dominant over the small y, that is our green. Okay, so basically in the dihybrid cross, we take 2 sets of characters. Okay, let's say the seed might be round yellow, understood, and the seed might be green and wrinkled. Okay, green and wrinkled. Okay, so what will be the gametes produced here? From these 2, okay, from these 2 the capital R will be produced, from these 2 small capital Y will be produced. So the gamete produced will be capital R, capital Y. Okay, in this case what will be the gametes produced? From these 2, small r, one smaller comes, and small y, that is one small y comes. Okay, so in the first filial generation the plant will be capital R, capital Y, small y, that is round and yellow. Okay, round and yellow. Okay, now we are doing the self pollination. Okay, we are doing the self pollination here. Okay, we are doing the self pollination here. So what happens now? Now we will make a Punnett square. Okay, let's make gametes first. Okay, for making gametes you will take combination of alleles. Okay, कैसे? Let's say तुमने यहाँ से एक capital R उठाया, यहाँ से एक capital Y उठाया, that will be your one gamete. यहाँ से तुमने capital R उठाया, यहाँ से तुमने small y उठाया, तो that will be another gamete. यहाँ से तुमने small r उठाया, यहाँ से तुमने capital Y उठाया, तो that will be another gamete. And the last one will be small r and small y. तो हम देखते हैं कि लाइक वो सारे एलील खुद के बीच में डिफरेंट तरीके से खुद में वो कॉम्बिनेशन क्रिएट करते हैं. सो दिस इज द थर्ड लॉ ऑफ मेंडल, दैट इज द लॉ ऑफ इंडिपेंडेंट असॉर्टमेंट. सो दिस इज द लॉ ऑफ इंडिपेंडेंट असॉर्टमेंट. ओके, सो दिस इज द लॉ ऑफ इंडिपेंडेंट असॉर्टमेंट. नाउ व्हाट विल बी हैपन? We will make a Punnett square like this. Okay, we will making a Punnett square like this. 1 2 3 4, 1 2 3 4, capital R, capital Y, capital R, small y, capital small r, capital Y, small r, small y. capital R, capital Y, capital R, small r, capital Y, small r, small y. Okay. Now we are writing the answers. Okay. Capital R, capital R, capital Y, capital Y. Capital R, capital R, capital Y, small y. Capital R, small r, capital Y, capital Y. Capital R, small r, capital Y, small y. Okay. So like this we will fill up the table. Small r, capital Y, small y. Capital R, small r, capital Y, small y. Capital R, small r, small y, small y. Capital R, small r, capital Y, small y. Small r, small y, small y. Okay. So this gives us the like whole thing of the dihybrid cross. तो इससे हमको क्या मिलेगा? See this table. Okay. See this table. So what will be the phenotypes of the plants? That will be round yellow. These are the round yellow. 1, 2, 3, 4, 5, 6, 7, 8, 9. These are the 9 yellow. Okay, 9 round and yellow. 3 round and green. That is this one, round and green. 1, 2, 3. 3 wrinkled yellow. 1, 2, 3. Okay, and the 1 wrinkled green. That one is this one. So this is a very famous question of Neet. Like what is the phenotypic ratio in case of a dihybrid cross? Okay, so your answer will be 9:3:3:1. Okay, so the answer will be 9:3:3:1. Okay, and what will be the genotypic ratio? That you might remember, that is 1:2:2:1:1:2:1. You can just remember this one. Okay, just keep it like a phone number. तुम जैसे phone number याद करते हो, इसको भी याद कर लो. वह 1 2 2 4 1 2 1 2 1. Okay, so this gives the idea of a dihybrid cross. Okay, so let's take a question. Monohybrid cross. Okay, so monohybrid cross का क्या होता है? क्या होता है उसका phenotypic ratio? Let's say उसका होता है okay, let's say test cross. So test cross में क्या होता है? That is 1:1. Okay, so D का 1 कहाँ पे देखा? D का 1 ये भी होगा, ये भी होगा, ये भी होगा. Okay, तो let's look at another options. Incomplete dominance. Incomplete dominance हमने क्या बोला था? Genotypic and phenotypic ratio will be same. That is your 1:2:1. So C का होगा 2, C का होगा 2, C का होगा C का होगा 2, C का होगा 2. तो ये वाला ऑप्शन होगा. C का 2, D का 1, 1 का 3, B का 4. So this will be your answer. So this will be your answer. Okay. A का 3, that is monohybrid cross. That is 3:1. It's phenotypic ratio. Then it's come B का 4, dihybrid cross. That will be 9:3:3:1. Then that is 2. That is 1:2:1. Then 1:1. Okay. This was asked in Neet 2023. What is the expected percentage of a F2 progeny with a yellow and inflated pod in dihybrid cross? Experiment involving pea plants with green colored inflated pod and yellow colored constricted pod. So they are basically saying कि like ये वाला cross का result क्या होगा, right? We have done it like here, and they are saying कि like the with yellow and inflated pod, yellow and inflated, right? So yellow होता है तुम्हारा dominant, right? So yellow is dominant. Yellow is dominant. Okay, and the inflated pod is also dominant. Okay, so what is the percentage that will be yellow and inflated? Okay, so one inflated one is recessive. Okay, recessive. So what is the, I said the ratio 9:3:3:1. So this will be both dominant, but here one is dominant and another one is recessive. So this will not be your answer. So your answer will be 3. So 3 by all plus, that is 16. That gives us near about 18.75%. So your answer will be 18.75%. Okay. So as I discussed कि law of independent assortment क्या होता है, right? कि क्या होता है कि like all the genes and all the alleles, they can combine with each other in different combinations. So that is our law of independent assortment. Okay. The next topic, that is chromosomal theory of inheritance. So let's say कि it was remained unrecognized till 1900, this theory. Okay, firstly, wait a minute, communication was not easy because Mendel का काम जो हुआ था तब communication उतना easy नहीं था. In those days his work could not be widely publicized and his concept of genes as stable discrete units that controlled his expressions of traits was not accepted. Okay, तो मेंडल का काम एक्सेप्ट नहीं किया गया था. उसके बाद क्या हुआ था? In 1900, three scientists, there are Devries, Correns and von Schermark. They like reframed the Mendel's work. Okay. So this discovered to the structures. तो actually जब माइक्रोस्कोप डिस्कवर हुआ था तब डिफरेंट लाइक स्ट्रक्चर्स लाइक न्यूक्लियस, सेल मेम्ब्रेन और छोटे छोटे ऑर्गेनेल्स वो लाइक हम उसको डिस्कवर कर सकते थे, राइट. तो बेसिकली क्या होता है, वाल्टर सटन एंड द थियोडोर बोवेरी नोटेड दैट द बिहेवियर ऑफ अ क्रोमोसोम वाज पैरेलल टू द बिहेवियर ऑफ अ जीन एंड यूज्ड क्रोमोसोम इन्वॉल्वमेंट. सो हु वर द साइंटिस्ट हु डिस्कवर्ड द क्रोमोसोमल थियोरी ऑफ इनहेरिटेंस? दैट वर द वाल्टर सटन एंड द थियोडोर बोवेरी. दैट वाज द वाल्टर सटन एंड द थियोडोर बोवेरी. ओके. Let's solve these questions. These are not too much hard. See, in antirrhinave snapdragon, a red flower was crossed with a white flower in F1 generation. Pink flowers were obtained. When pink flowers are selfed, the F2 generation showed white, red and pink flowers. Choose the incorrect from the following. Pink flower in F1 is due to incomplete dominance. सही बोला है, because snapdragon is the example of an incomplete dominance, right? The ratio of F2 is 1:4, 1/4 red, 2/4 pink, 1/4 white. ये भी सही है. Law of segregation doesn't apply in this experiment. ये गलत है. Law of segregation is applied. Which doesn't apply? That is law of dominance. The experiment doesn't follow the principle of law of dominance. तो ये सही है. तो what will be the answer? It will be law of segregation doesn't apply in this experiment. Okay. Next question. A certain plant homozygous for yellow seeds and red flowers crossed with a plant homozygous for green seeds and white flowers. The F1 plants had a yellow seeds and a pink flowers. Okay. The F1 plants were selfed to get F2 progeny. Assuming independent assortment of the two characters, how many phenotypic categories are expected? Okay. So what are the phenotypic categories? That is yellow, yellow wala seed, that is yellow seed, right? Yellow seed, and that is green seed. Okay, that is green seed. But in the color there are three colors, right? Capital R is red, capital R small r is pink, and the small r is white. Okay. So how many types of phenotypic characters? That will be your here 2 multiplied by here 3. So your answer will be 6. Like one will be yellow red, one will be yellow pink, yellow white, another one will be green red, green pink and green white. So there will be total 6 combinations of characters phenotypic. Okay. Okay. The chromosomal theory of inheritance was proposed by Sutton and Boveri. Okay, this one is a very important question. This has been asked many times in the NEET exams. Okay. Thomas Hunt Morgan and his colleagues led to discovering the basis of the variation that sexual reproduction produced. Morgan worked with his tiny fruit flies, that is Drosophila melanogaster. That is an important question. The animal and the insect that was used by the Morgan and his colleagues, that is the Drosophila melanogaster. Okay, Drosophila melanogaster. Okay. Why it was used? There are two reasons. They complete their life cycle about 2 weeks, and a single mating could produce a large number of progeny lies. Okay, flies. So and the female and the male flies were easily distinguishable. Okay, so this was the reason they used Drosophila as their the Drosophila used for their work. Okay. Now coming the linkage and recombination. Okay. So what is linkage and what is recombination? Basically, linkage is two closely adherent characters. Okay. Two closely adherent characters that cannot segregate during the process of recombination. Okay. Two characters that cannot being separated during the process of recombination. तो क्या होता है? The two characters and the two genes that are inherited to the next generation very frequently they are inherited together. Very frequently they are inherited together. Okay. So this is the called linkage. For example, Morgan hybridized yellow-bodied white-eyed female to brown-bodied red-eyed females and intercrossed their F1 progeny. He observed that the two genes didn't segregate independently each other and the F2 ratio deviated very significantly from the 9:3:3:1. Okay, so this was the mainly linkage. Okay, so in the diagram you can see in this case, okay, in the cross A you can see yellow, white and wild type of flies, they were being crossed together, and 98.7% were parental types and the recombinant types were 1.3%. Okay, so this gives us a brief idea कि parental वाला part ज़्यादा हो रहा है, so here linkage is more. Okay, linkage is more stronger, right? Because linkage is more stronger, so they are not being separated and the product is similar to the of the parent. Okay, so the next portion, that is the cross B, that is white miniature fly that is being crossed with a wild type. Okay, so that gives 62.8% parental and 37.2% recombinant type. So these percentage questions were frequently asked in NEET exams. Okay, you have to remember at least this percentage, that is 98.7%, and this percentage 62.8%. Okay, these were being asked many times in NEET exams. Okay, so from this portion the main topics have been already covered. Okay, so now we can move to the solving some questions. Okay, let's see. The chromosomal theory of inheritance was proposed by, it was proposed by Sutton and Boveri. It was proposed by Sutton and Boveri, and it was experimentally done by Morgan and his colleagues. Okay, it was experimentally done by Morgan and his colleagues. Okay, what map unit is adopted in the construction of a genetic map? तो इसका यूनिट होता है सेंटीमॉर्गन. सो दैट इज डिस्टेंस बिटवीन two genes on a chromosome representing 1% crossover. 1% crossover. So that is a centimorgan. That is a centimorgan. Okay, remember that. This one is not given in Ncert. This was an old question that was from NEET 2019. Hoping like these types of question can't come in recent papers 2025 and ahead one. Okay. Let's say. Frequency of recombination between gene pairs on same chromosome as a measure of distance between genes to map their position on chromosome was used for first time by, it was used by what I said, that was Thomas Hunt Morgan. Like Morgan इसको experimentally demonstrate किया, and the Sutton and Boveri, they proposed the theory, and Morgan himself proved the theory by his explanation and his experiments. Okay. Now, what is a polygenic inheritance? See, you can see in the name, it is called a polygenic inheritance. That means one character is controlled by different genes. Example is a human skin color, human height. Okay. See, there are many characters in a human being or in many plants. Okay, that is not too much distinct. Okay, distinct means, let's say, in humans, people are tall, right? People are tall, they are 6 feet 5 inches. There are short people too, like there are 4 feet 8 inches people, 4 feet 9 inches people, right? But there are people like 5 feet 5 inches, 6 inches, 7 inches. So there is not basically a dominant one and a recessive one. Okay, so the whole thing is like a gradual decrease or increase. Okay, so what they have said, there are some characters, there are some traits which are not so much distinct in their occurrence and are spread across a gradient. That is a gradient, right? That is a gradient. For example, in humans, we don't just have a tall or a short people, two distinct human alternative, but a whole range of possible heights. As I said, it can be 6 feet, it can be 5 feet 9 inches. Okay, it is a spectrum or a gradient. Okay. Such traits are generally controlled by 3 or more genes and thus called polygenic traits. They are called polygenic traits. Okay. Besides the involvement of the multiple genes, polygenic inheritance also takes in account into the influence of the environment. So another example is human skin color. Okay, so remember कि polygenic inheritance का example क्या होगा? Human skin color, human height. Okay, that is mainly asked in the NEET examination, and you have to remember this one. Okay, like human skin color is an example of pleiotropy, polygenic inheritance, incomplete dominance. So answer will be polygenic inheritance. Okay, pleiotropy. So basically प्लियोट्रॉपी मतलब एक जीन वो बहुत सारे कैरेक्टर्स को कंट्रोल करेगा. इन पॉलीजेनिक इनहेरिटेंस वन कैरेक्टर वाज कंट्रोल्ड by different genes, and here one gene controls different characters, as I have written here. Okay, one gene that controls different characters and many genes that controls only one character. Okay, so that is polygenic inheritance, and this one is pleiotropy. Okay, so what is an example of a pleiotropy? Pleiotropy example will be phenylketonuria. So what is phenylketonuria? Phenylketonuria is a inborn metabolism disorder of a child. Okay, it is a inborn metabolism disorder of a child. तो इसमें क्या होता है? Like there is a mutation in the gene that codes for the enzyme phenylalanine hydroxylase. That is phenylalanine hydroxylase. So in pleiotropy, what you have to remember? You have to remember the example, that is phenylketonuria. Phenylketonuria is done by the deficiency of which enzyme? Phenylalanine hydroxylase, and there is a mutation in the gene. Okay, you have to remember these three things. Okay. Now coming to the sex determination. Okay, तो sex determination कैसे होता है? In humans we know we have homogametic females and the heterogametic males. That means the homogametic, that means XX are females and XY are males. Okay, XY are males. Okay, so what we can see, there is a question, we can solve this. Given below two statements. When many alleles of a gene govern a character, it is called polygenic inheritance. When many alleles of a single gene govern single character. Okay, when many alleles of a single gene govern a character, that is called polygenic inheritance. This is a wrong statement, right? This is pleiotropy. And see this one, in polygenic inheritance, the effect of each allele is additive. This one is right. And when many alleles of a single gene govern a character, it is called polygenic. This is wrong. So your answer will be 1 is false, 2 is true. Okay. In a marriage between male of blood group A and a female with blood group B, the progeny had either blood group AB or B. What could be the possible genotype of the parents? So what they are saying ki, let's say one is A blood group, it might be IA or IA-I0, right? IA-I0, right? And the father is and the female is B. Okay, IB or IB-I0. Okay, and what they have said कि they have both A and B. Okay, so what could be the possible genotype? Okay, so genotype might be one should be of O, okay, and another should be of O. It might be O or it might be this one. So we can see here, this might be your answer. Okay, this will not be your answer because here B will not be present. All will be AB. In this question, this one is also not the answer. All will be B. All blood groups will be B. And in this portion, all will be AB. So what is your answer? This one. Male is this one and female is this one. Okay. A mother has a A positive blood group, the father has a B positive and the child is A positive. What can be the possibility of genotypes of all the three respectively? Let's say mother का है A positive blood group. Okay, mother is A positive, father has B positive and the child is A positive. So you have to understand that कि mother का भी A है, father का भी B है. So one of them need to be have a IO gene. That means IO, right? So IB answer can be an answer. IBI can be answer and IBI it is also answer. Okay. Okay, no, the child is A positive, so this can't be answer. So child ये होगा, ये A है child का, ये भी A है child का, ये भी A है child का, right? So अब see, let's see. From this one, the child is A positive, so this can happen. This can't happen. All will be AB, and this can't happen. So P or D, B or E, B or E होगा, right? So answer will be B and E. Okay, B and E. Wait, answer will be B and E. Okay. So how is determined? Sex is determined in humans. males are XY, females are XX. Okay. In Drosophila, the same, females are XX and males are XY. Okay. But in the like birds, okay, in the birds, males are homogametic, that is ZZ, and females are heterogametic. So the gender of the child depends upon the male in humans, but in the birds it depends upon the female, because female is heterogametic. Okay. The experimental verification of the chromosomal theory of inheritance was done by, this was done by Sutton and Boveri, right? Sorry, this was done by Morgan, right? This was done by Morgan. Experimental, that is done by Morgan. Okay. So sex determination in human beings. So let's say ki the female is XX and the male is XY. Okay. So what will be the gametes? That will be X and X in this case, and X and Y in this case. Okay. So just made a Punnett square. Okay, like this. That is XX XY. So XX hoga, XX hoga, XY hoga, XY hoga. So there is a 50% chance that a male child or a female child can be produced. Okay. Or genetic makeup of the sperm that determines the sex of the child. So the sex of the child is dependent upon the father, not the mother. Okay. In our society, mothers are being treated very bad for giving birth to the female child. But it's not true. The whole genetic makeup is mainly dependent upon the papas, mean father's chromosome variation. Okay. Now sex determination honeybee. So in honeybee, it's not like that one, like unka X or Y chromosome hai. So number of sets of chromosome individual is dependent. Okay. Let's say female ka are diploid and males are haploid. Okay. Males produce gametes by the mitosis. So in male honeybee, male produce gametes by the process mitosis. Okay. But in females, they produce it by meiosis. Okay. And gametes are 16 and 16, and 16 will produce one male. Okay. So the females are diploid and males are haploid. Okay. So males produce gametes by mitosis and females produce gametes by meiosis. XO type of sex determination can be found in. XO type can be found in. This is a direct question from Ncert. See this one. Large number of insects, that mechanism of the sex determination is XO type. Okay. So XO type is mainly found in which? That is grasshopper. It was asked in Neet 2022. So the answer will be grasshopper. So XO will be of the grasshopper. Okay. Answer will be grasshopper. XX XO method determination syndrome. ZZ ZW method of sex determination, that is mainly in the female heterogametes, right? So D ka hoga 2. D ka hoga 2. There is only one option, that is D ka 2. Okay. Mutation. Basically mutation is nothing. Mutation is like change in one single base pair. It might be point mutation, it might be frame shape mutation. But here we have to remember this portion, like for the mutation only classical examples are the sickle cell anemia. Okay. So it happens due to deletions and insertion of base pairs. Okay. The topic mutation will be covered in the next portion, that is the molecular basis of inheritance. Stay with us and we will finish the whole syllabus in just some few days. Okay. Now we are coming to the most important topic of the chapter, that is the genetic disorders and the pedigree analysis. Okay. So what we can do, we have to remember this portion. Okay. So in a pedigree chart, what we can do, we can find the type of a disease. Okay. From the history of a family disease, we can find the type of a disease. So what a pedigree chart have, the box one are male, the rounded ones are female, the triangle, this shape is sex unspecified. These are the affected. The darker one, they are affected. This is mating, that means marriage. This is consanguineous marriage, that means marriage between brother, sister, or any close relative. Okay. These are the like the generation, and these are the parents with affected child. Okay. So so हम pedigree को कैसे analyze करेंगे? Let's take a pedigree. Okay. Let's see the we have to find the number of generations. Okay. How to analyze a pedigree? We have to find the number of generation. This is the first generation. Okay. This one is the second generation. This one is the third generation. This one is a fourth generation. Okay. This one is a fourth generation. Okay. Sorry. This one is a third generation. My mistake. These both are third generation. Can you see? This is also third generation. This is a third generation. Okay, this one is third generation. So we have to just first find the number of generations. Okay, then we have to see we can find the disease in all the generations, or is there a generation gap? So we can see here is no generational gap. Okay, so if there is no generational gap, that signifies the disease is a dominant disease. Okay, disease is a dominant disease. Okay, and if you see any gender bias, have you seen any gender bias? Like, see, here is a female affected, here is a male affected, female affected, female affected. So there is no gender bias. So if there is no gender bias, that means it is a autosomal disease. This is a autosomal disease. Okay, this is a autosomal disease. So you can see here, this is a autosomal dominant disease. That is example myotonic dystrophy. This question was asked in Neet 2022. This was asked in Neet 2022. To give an example of a autosomal dominant disease, the answer would be myotonic dystrophy. Myotonic dystrophy. So what is? Can you explain this one? See, this one is first generation. This one is second generation. This one is third generation. Okay. Do you see any generation gap? Yes, sir. No affected child, but affected. There is a generation gap, so it will be recessive. Okay. There is generation gap, that will be recessive. Do you see gender bias? Yes, no. Male, female, male. So there is no gender bias. So it will be autosomal recessive. So can you just you can just see a pedigree. You will see which type of disease it is. It is as much easy as that. You have to see the generation, first generation, second generation, third generation. Then you have to see there is a generation gap, that will be recessive. If there is no generation gap, it will be dominant. And third, if there is a gender bias, that will be something like sex chromosomal, it might be X or Y. If there is no gender bias, like all females, males are affected, so it will be recessive. It will be autosomal. Okay, it is as simple as that. So what is example of autosomal recessive disease? That is your sickle cell anemia. Okay. Now coming to the next point, that is color blindness. Okay. So what you have to remember, color blindness is a X-linked recessive disorder. This is a X-linked recessive disorder. Okay, this is due to the actually in the color blindness, people can't see the colors, like they can't differentiate the color red, blue, green. According to their name, they are called protanopia, deuteranopia, okay, tritanopia. That is not in scope of a discussion. Okay, so color blindness is a X-linked recessive disorder, and more males are affected in the more than the females. Why? Because males are the heterogametic, right? They have one X chromosome, one Y chromosome. If there is one defective X chromosome, let's say this is a defective X chromosome, the male will be easily affected. But in the case of females, there are two X chromosomes. So if one is affected, another might be normal. So it will be carrier. But and there is a less chance of being affected. So males are more affected than the females. Okay. See. Which of the following symbols represent mating between the relatives in the human pedigree analysis? That means consanguineous marriage. Okay. Consanguineous marriage. That is this one. First wala. Consanguineous marriage. Okay. Which of the following occurs due to the autosomal linked dominant trait? So what is your answer? Autosomal dominant myotonic dystrophy. Okay. Next part, hemophilia. Okay, hemophilia also a X-linked recessive disorder. Okay, hemophilia is also X-linked recessive disorder. So how can we find if a child is affected or not? Let's see. Thus, in the case of X-linked recessive disorders, the chart will be same. So I am explaining only the hemophilia, and you can do the color blindness on your own. Okay. Let's say there is a normal mother. Okay, normal mother. So what will be her genotype? That will be X capital H. Okay, capital H mane that is normal. Okay, and there is a hemophilic father. Okay, hemophilic father. Okay, so what we can do? What is the genotype? That is small h y. That is hemophilic. So what will be the gametes? We can draw the Punnett square like this. That is XH. It is X small h y. Okay, so this is a carrier, this is normal, and this is also normal. Okay, so you can see here only two carrier. This is a carrier, this is a carrier. So two carrier will be produced and no child will be hemophilic. So like this you can do some questions on your own. Okay, it is same in hemophilia and color blindness. Okay, so I am giving you a question. Okay, just solve it and write the answers in the comment box. Okay, there is a hemophilic mother. Okay, hemophilic mother. Okay, that marries a normal father. Okay, so what will be the chance of percentage affecting of the child? Okay, write your answers in the comment box below. Okay, the next portion, that is your sickle cell anemia. Okay, so sickle cell anemia basically is a autosomal recessive disorder. Okay, sickle cell anemia is mainly controlled by a pair of genes, that is HbA, HbS. Okay, mainly HbA is the normal gene and HbS is the sickled gene. Okay, so HbS will be a sickle cell anemic. Okay, and HbA, HbS will be a sickle cell carrier. Okay, and HbA, HbA is quite normal. Okay, HbA is normal. Okay, so what is the problem in the sickle cell anemia? Okay, the problem is here, this one. What is the problem? The defect is caused by the substitution of the glutamic acid by valine at the 6th position of the beta globin chain of the hemoglobin molecule. You have to remember it line by line. Okay, substitution of glutamic acid by valine at the 6th position of the beta globin chain of the hemoglobin molecule. This has been asked many times. Okay, and the gene is changed from GAG to GUG. GAG turns into GUG. Okay, then comes phenylketonuria. You know, it is an autosomal recessive disorder. Okay, this is an inborn error of metabolism, and this is the deficiency of a phenylalanine hydroxylase, as we discussed earlier. Okay, this is the deficiency of the phenylalanine hydroxylase. Thalassemia is also a autosomal recessive disorder. Okay, thalassemia is an autosomal recessive disorder. Thalassemia is basically of two types. One is alpha thalassemia, one is beta thalassemia. Alpha has 4 chains, beta has 2 chains. Okay, if 4 of the genes are defected, that is called alpha thalassemia major, and if 2 of the both beta chains are defected, those are called beta thal major. Okay, those are called beta thal major. Okay, so let's go to the important portion, that is your chromosome number. Okay, so beta globin chain is present on the chromosome number 11. This has been asked many times. And the alpha chain is present on the 16th chromosome, that is present on the chromosome 16. This one is present on the chromosome number 16. Okay, now coming to the chromosomal disorders. Okay, so what is a polyploidy and what is a aneuploidy? Aneuploidy means failure of segregation of chromatids during cell division. Cell cycle results in the gain of the loss of the chromosomes. It is called aneuploidy. Okay, if gain happens, that is called polyploidy. Let's say during segregation, 23 chromosomes being segregated, that is 22 plus X, and there is 22 plus Y. Okay, but during segregation there is a problem. Okay, it happens like this: 21 plus X, and it happens 23 plus Y. Okay, so here is a decrease in number, that is called aneuploidy, and here is increase in number, that is called polyploidy. Okay, so those are mainly complementary to each other. Okay, see, 2 or more alternative forms of gene, they are called allele, right? They are called allele. So 1 का 3, ये वाला आंसर होगा. Very easy. You can check the other options. B का 4. Cross with F1 progeny with homozygous recessive parent, that is called test cross, right? That is called test cross. Cross of F1 progeny with any of the parents, that is called back cross. And number of chromosome sets in plant, that is ploidy. Okay, that is ploidy. A normal girl whose mother is hemophilic, right? A normal girl whose mother is hemophilic marries a male with no ancestral history of hemophilia. What will be the possible phenotypes of the offsprings? Hemophilic son and hemophilic daughter. अच्छा, इसको एक बार करते हैं. Okay, let's do it once. अच्छा, a normal girl whose mother is hemophilic. So mother is hemophilic like this. Her mother is hemophilic, right? her mother is hemophilic, like this. Marries a male with no ancestral history of hemophilia, so the male will also be normal. Okay, so what will be the genes? That will be X small h, and it will be X capital H, Y. Okay, now just draw a Punnett square. Okay, just draw a Punnett square. X small h, X capital H, Y. Okay, so X small h, X capital H, X capital H, X small h, Y, X small h, Y. So what will happen? You see, females are carrier, so the females are carrier, right? So the females are carrier, and the males are affected. So what will be your answer? Carrier female and the carrier female. What will be the answer? Hemophilic son होगा, this one, hemophilic son and a hemophilic daughter. Okay, A, and B, hemophilic son, carrier daughter, this one, hemophilic son or carrier daughter. Okay, B. Okay, this one is the answer. Okay, see, sickle cell anemia and hemophilia are autosomal dominant traits. No, they are not. Sickle cell anemia is autosomal recessive and hemophilia is X-linked recessive. So this statement गलत है. Sickle cell anemia and hemophilia are disorders of the blood. ये सही है. This is a true. So 1 is correct, 2 is wrong. So option D. Option A, sorry. Statement 1 is incorrect and statement 2 is correct. Okay. तो क्रोमोसोमल डिसऑर्डर में क्या क्या पढ़ना पड़ेगा हमको? डाउन सिंड्रोम, देन क्लाइनफिल्टर सिंड्रोम, देन टर्नर सिंड्रोम. सो व्हाट वी नीड टू रिमेंबर फ्रॉम दिस पोर्शंस, डाउन सिंड्रोम में क्या होता है? इसको बोलते हैं ट्राइसोमी 21. द नंबर ऑफ 21 क्रोमोसोम इज 3. ओके, व्हाट इज द मेन फीचर? दैट इज पामर क्रीज. इट हैज बीन आस्क्ड मेनी क्वेश्चंस. Like in the symptoms, they have given you your palmar crease, congenital heart disease, so you have to diagnose it with a Down syndrome. Okay, this is a Down syndrome. So what is a Klinefelter syndrome? That is extra X chromosome in a male. Okay, so that is 47,XXY. What is the genetic karyotyping? That is 47,XXY. So this is more important, 47,XXY. This has been asked many times. What is the karyotyping of the Klinefelter syndrome person? Okay. And what is Turner syndrome? Turner syndrome is basically aneuploidy and 45 with XO. So इसका क्या होता है karyotyping? 45, XO. So here is a lack of X chromosome in a female. Okay. So what are the ovaries are rudimentary. So Turner syndrome का जो होता है, she is biologically a female, right? And Klinefelter, the person is a biological male. Okay. This is a big important question that has been asked few times in AIMS exams. Like what is the biological gender of the Klinefelter syndrome patient? He will be male, and the Turner syndrome, the patient will be biologically female. Okay. Broad palm with a single palm crease is visible in a person suffering from. जो मैंने बोला था, palm crease में क्या बोलोगे? That is Down syndrome. In a female individual with a small round head, furrowed tongue, partially open mouth, and broad palm with characteristic palmar crease. You see the word palmar crease. You just mark the answer Down syndrome. That is trisomy 21. Okay. Which of the following statements are correct about Klinefelter syndrome? It was first described by Langdon Down. No, he described Down syndrome. Such individual has a overall masculine development, however the feminine development is also expressed. In the Klinefelter syndrome, this is true, right? The affected individual is short-statured. Physical, psychomotor, mental development is retarded. This is wrong. Such individual are sterile. This is true. So answer will be B and E. The answer will be B and E. Okay. This has been asked in Neet 2023. Next question. Which genetic condition each of the cell affected person has 3 sex chromosomes XXY? So what I said, XXY means Klinefelter syndrome. In which disorder change of single base pair of the gene for beta globin chain results in change of glutamic acid to valine. जब देखोगे ग्लूटामिक एसिड और वैलिन वाला पोर्शन, यू विल राइट द आंसर सिकल सेल एनीमिया. व्हाट विल बी आंसर? सिकल सेल एनीमिया. आग बंद करके सिकल सेल एनीमिया. ओके, सिर्फ करेक्ट मैच. फिनाइलकीटोन्यूरिया ऑटोसोमल डोमिनेंट ट्रेट, गलत है, ये रिसेसिव है. सिकल सेल एनीमिया ऑटोसोमल रिसेसिव ट्रेट क्रोमोसोम 11, गलत है, ये थैलेसीमिया होगा. सो थैलेसीमिया एक्स लिंक्ड, ये भी गलत है. हिमोफीलिया वाई लिंक्ड, ये भी गलत है, हिमोफीलिया एक्स लिंक्ड है. सो this will be correct. Sickle cell anemia, autosomal recessive trait, and it is a fault in a beta globin chain, right? So beta globin chain का क्रोमोसोम कहाँ पे रहता है? बीटा ग्लोबिन चेन का क्रोमोसोम रहता है क्रोमोसोम 11 पे. Okay. In a cross between a male and female, both heterozygous for sickle cell anemia gene, what percentage of progeny will be diseased? So male और female both heterozygous for the sickle cell anemia. So heterozygous means ऐसे होगा. That be like this. That will be crossed like HBA and HBS. So now make a Punnett square. It is quite easy for you guys now. Capital A is, I am writing like this, AS. That is normal. That is carrier. That is carrier. That is sickled. So sickled will be 25%. 1 by 4. That is 1 by 4. That is 25%. Okay. That is 1 by 4. That is 25%. If a colorblind female marries a man whose mother was also colorblind, what are the chances of her progeny having colorblindness? तो यहाँ पे क्या बोला है? फीमेल कलर ब्लाइंड है, राइट? ए मैन जिसका मदर भी कलर ब्लाइंड था, तो वी कैन सी अ पैटर्न दैट इज कॉल्ड क्रिस क्रॉस इनहेरिटेंस, राइट? क्रिस क्रॉस इनहेरिटेंस. इसमें क्या होता है कि कोई एक्स लिंक्ड रिसेसिव डिसऑर्डर होता है दैट कैरीज फ्रॉम मदर टू देयर सन एंड फ्रॉम देयर फादर टू देयर डॉटर, लाइक क्रिस क्रॉस इनहेरिटेंस. तो लाइक दे हैव सेड कि लाइक दिस इज हीमोफीलिया एंड दिस इज कलर ब्लाइंडनेस, तो इट विल बी पास फ्रॉम अ फादर टू हर मदर, राइट? मतलब किसी की मम्मी से उसके बेटे को वो लाइक दैट विल बी पास्ड देयर. ओके, सो यहाँ पे बोलता है मैन हुज मदर वाज आल्सो कलर ब्लाइंड, सो द मैन विल आल्सो बी कलर ब्लाइंड. हियर इज द कैच ऑफ द क्वेश्चन. हियर इज द कैच ऑफ द क्वेश्चन. ओके, सो द मेल आल्सो बी विल बी द कलर ब्लाइंड. So what they have said, what is the chances of her progeny to be color blind? So now what we are doing a Punnett square. Okay, again a Punnett square. So x small c, x small c, x small c, y. Okay, so this is a color blind, this is a color blind, this is a color blind, this is a color blind. So all the progeny will be color blind. So answer will be 100%. Okay, so this finishes our lecture on the principles of inheritance and variation. So what were the main topics? That were the Mendel's law of inheritance. Okay, monohybrid cross, law of dominance, law of segregation, dihybrid cross, law of independent assortment, deviation from the Mendel's law, that is incomplete dominance, codominance, pleiotropy, polygenic inheritance, multiple allele. Okay, the blood grouping wala numericals they can give you in the exams. Okay, और क्या था? And the next portion was like in the chromosomal theory of inheritance that was proposed by Boveri and Sutton, and that was proved by the Morgan and his colleagues. Okay, then we discussed something about the the chromosomal disorders and the the chromosomal disorders and the genetic disorders. Okay, then we discussed about the sex determination in animals, man, and different animals, right? So you can see here the whole topic is covered within a video of 1 hour 20 minutes. Okay, hope this video will help you in your preparation, and I have tried to make the video too much concise ताकि तुमको ज्यादा टाइम ना देना पड़े इस चैप्टर के लिए. And I will also provide the PDF of the video in the linked description and in the pinned comment. Okay, and further, if you want to solve the DPPs of this module, give some tests of the module, please go and visit our website impetuslearning.in. We will provide you, or you can WhatsApp us on the number. Okay, you can WhatsApp us on our number. I can show you the number. Okay, this is our WhatsApp number 933042847. You can WhatsApp us on this number. You can connect us on the impetuslearning.in. Just वहाँ पे register कर दो. अगर मेरा पढ़ाया हुआ तुमको अच्छा लगा तो you can go there and ask for a personal one-to-one interaction. I will be interacting with you guys, and the whole session of the interaction and personal mentorship will be free before the NEET 2025. So go and grab the chance. Okay, and this was a long lecture of 1 hour 20 minutes. So go and have a cup of coffee or tea, and then just practice the DPPs and solve some MCQs or the PYQs. And if you have any doubts further, feel free to contact us on impetuslearning.in. Okay, thank you. Jai Hind.